Today's presentation is What's New in the Simcyp Simulator version 18. Our speakers today are Nikunjkumar Patel, Rachel Rose, and Shriram Pathak. Nikunj is a principal scientist at Certara, where he's involved in oral and dermal absorption modeling projects. Shriram Pathak is a senior research scientist at Certara, where he leads development and improvement of the Simcyp Simulator absorption and bioavailability models. And Rachel Rose is a principal scientist at Certara, where she works on developing and applying PBPK-PD models for large and small molecules. Rachel, Nikunj, and Shriram, welcome to today's event. I'll now turn it over to Nikunj to begin the presentation.
For the webinar on what's new in Simcyp version 18. So I'm going to take you through one of the items that we have incorporated in version 18, which is modeling of entero-hepatic recirculation of metabolite, and also metabolite to parent interconversion in the gut lumen, which will probably mostly driven by microbial flora. Yeah, so the new features for this project in version 18 involves the ability to model the luminal disposition of substrate as well as primary metabolite in terms of interconversion to each other or simply a loss in the lumen. You can model entero-hepatic recirculation for primary metabolite, and with that you would be able to use the features of the Adam model for primary metabolite, which Adam model is the advanced dissolution metabolism absorption model in Simcyp Simulator. And with that, you would be able to use the gut wall transporter as well as the metabolism within the gut wall for primary metabolite. And you can use the victim or perpetrator DDI for the metabolism as well as transport for the primary metabolite in the gut wall. So there are a lot more enhancements available to the primary metabolite as part of this project. Just going further into the luminal metabolism, there are, I think many of you are already aware, but there are significant number of phase 1 and phase 2 metabolic reactions that are already reported to be happening in the gut lumen, which is mainly driven by microbial flora or microbiota in the gut lumen. So there are some of them like reduction, hydrolysis. They are the most common reactions, but there are others like, for example, N-oxide cleavage that is responsible for ranitidine metabolism are also quite predominant. They are basically mostly deconjugation is for glucuronide. So if your glucuronide metabolite enters the gut lumen via entero-hepatic recirculation, they can convert back to the substrate and then get absorbed again and can enter this metabolite and parent interactive entero-hepatic recycling. And these are not just reported reactions, but they are also clinically meaningful. For example, for digoxin, it is already known that because of the degradation in the lumen, because digoxin is a narrow therapeutic index drug, it could affect the efficacy of the drug. And there are certain drugs like sorivudine, which I'm going to emphasize here as an example, where the luminal disposition could lead to safety concerns or toxicity. So this is an example of drug which is sorivudine, an antiviral drug that was introduced in Japan in early 1990s. Was withdrawn within the 40 days of approval because of lethal drug-drug interaction with anti-cancer drug tegafur, which is a prodrug of 5-fluorouracil, because it led to death of 18 patients. And the main reason was that the sorivudine is getting metabolized to BVU, which is the 2-bromovinyl uracil in the gut lumen via gut flora, and this metabolite actually gets absorbed in the system, and in the liver it irreversibly inhibits the enzyme DPD, which is responsible for the catabolism or the metabolism of or degradation of 5-fluorouracil. So when these drugs were combined, which was not tested in the clinical trials, so after approval, that led to toxic concentrations of 5-fluorouracil because it's no longer metabolized, and that led to a lethal DDI. Now the question here is that it took several years after the approval to establish the mechanism of toxicity because the luminal disposition is not routinely assessed within the drug development programs, and that had a severe consequences after the approval. So I think it's very challenging to have a full characterization of luminal disposition in a drug development program, but there has to be some experiments or some simulations to be done to understand the impact of luminal disposition on the efficacy and toxicity of new drugs. And rather than having as a post-approval thought or in very late stages of development, it needs to be incorporated in early stages. And I think combining in vitro experiments with PBPK could help this help bridge the gap. Why? Because if you look at the animals, there is a significant difference in terms of the microbiota or the luminal composition within animal and human species. So animals are a very poor surrogate to predict what might happen in the gut lumen of humans. As you can see here, the most common pathway or the most known pathway in the lumen is the beta-glucuronidase enzyme. And as you can see, the activity can be thousands of fold different between human and animals because the types of bacteria and the amount of bacteria across the GI tract, as you can see at the bottom figure, are significantly different in animal and human. And one of the reasons is because human eats more hygienic food as compared to animals. So as you can see in animals, you have some of the bacteria even in the stomach and upper intestine, while in human, which is represented as a black bar, dark black bar, you can see that there is, or in the healthy individuals or in healthy humans, there is no microbiota up to upper and even lower intestine. So the microbiota predominantly starts in human from lower intestine and especially in colon. So animals are a very poor surrogate. So I think then there is a way to rely on in vitro experiment and scale it to human condition via PBPK. But then there are some challenges, especially for luminal metabolism, because there is a very small amount of information available. So the first question is that, is the enzyme secreted? So here the blue and the purple boxes indicate different species of bacteria in the gut lumen. Because these different species can produce the same enzyme, so we don't know what is the correlation between bacteria and the enzyme. The second question is that, is the drug enter bacteria to get metabolized, or the bacteria secretes enzyme in the lumen to interact with the drug? We did some literature search, really extensive, but we couldn't find a concrete evidence what's happening. But most of the likely explanation is that the enzyme is secreted outside and then interacts with the drug. Also, there is some evidence that the drug can induce its own metabolism because once the drug once the compound is in the system, it induces more enzyme to be produced by microbiota, and then it can have a positive feedback on its own metabolism. So there are some open questions. The other one is that we don't know enough about the distribution of microbiota across the GI tract and its population variability, because microbial composition strongly depends on the diet, the lifestyle, and also there are some other environmental factors involved. So it's much more complex to predict inter-individual variability of the luminal disposition. The final challenge is that there are no standard in vitro experiments. For example, for liver, we have a standardized in vitro experiments where you can have like hepatocytes, or you can have a recombinant system, and we have developed suitable scalars to be able to extrapolate quantitatively. But for luminal metabolism, there are no standard experiments available. While we know anaerobic condition, osmolarity, the pH, the nutritional status of the fecal homogeneate experiments are important. But most of the time the fecal homogeneate collected are diluted, and we don't know if it has any consequences on the viability of the bacteria or the ability of bacteria to produce the enzyme. So there are many gaps here. But with that, still we implemented the model in simulator, which is general enough that user would be able to parameterize it for practical use based on the data they have. So as you can see on the screen, on the substrate screen or the drug or parent screen, you can You can see the new gut lumen screen where you can define the luminal metabolism as intrinsic clearance per gram of content, and then that is scaled to the total gram content in each segment of the GI tract. And when you tick this button below the MET1, so that means that whatever the substrate is metabolite is converted to metabolite. If you don't activate it, that means it's just a simple loss or degradation. You can account for in vitro binding if you have known in your experiment, or you can also select different reference segment and scale it to the relative activity across the GI tract. And it's the same for the metabolite, so you can convert the metabolite back in the lumen and you can select any segment. So here, for example, this is an example of a prodrug which is predominantly converted to metabolite in the stomach by pH-based degradation. Or you can have a beta-glucuronide metabolite that gets converted to the parent, especially in the colon via microbial flora. So you can basically account for them either single individual pathway or together. So overall, I think this is a very busy figure, but I hope it gives you the picture of the total additions that have been done as part of this project in version 18. So all the blue lines indicate the new processes or pathways introduced. So now, for example, from version 18, you can convert the substrate to metabolite or metabolite back to substrate from stomach up to feces compartment. And whatever PM1 is formed in the lumen, because it can use the ADA model or advanced absorption model within simulator. So it can be absorbed or it can undergo efflux and uptake transport. Once the primary metabolite is in the gut wall, it can be further metabolized to secondary metabolite, and again it can enter the system. Once the drug is in the liver or the metabolite is in the liver, now it can also get secreted in the bile and can get re-enter the system by entero-hepatic recirculation. So it's allowing more complicated scenarios to be simulated in version 18 if it is relevant to your drug or metabolite. However, as I already indicated, there are few gaps that we need to fill. First of all, we have implemented this model and we are verifying the model in terms of its applicability for prodrug simulations as well as the conversion of metabolite back to parent in the lumen. So we are also looking for data. So if you have data or problem at hand, we would be really happy to work with you. We are also looking to collaborate with the lab experts, because as I already indicated, there are no standardized in vitro experiments. So I think unless we have a standardized in vitro experiments, it's very challenging to have a well-established in vitro in vivo extrapolation, because if the in vitro experiment is not standardized, it's very challenging to see what type of scaler to use. Also, there is a huge potential of having a more mechanistic extrapolation if we know the microbial composition and abundances across the GI tract within the population. So I think we would be able to scale it appropriately for different populations and individual subjects' risk. With that, I would like to acknowledge people involved, so Sibila and Masood, Mian, Matt, Ramki, and Janak. So these all people contributed in this project. And we will take the questions in the end. So if you have any questions, you can put it to the chat, or maybe you can hold on until end. So now I will pass it to my colleague Rachel, who is going to take us through the new tumor models in version 18.
Thank you, Nikunj. Just mentioned, I'll take you through the new features in the version 18 of the simulator for the development of tumor models. So just some background first. So I'm sure as many of you are aware, cancer is a major cause of death globally, so it's a really active area of interest for drug development. However, there are challenges and quite a lot of failures in the clinical development phase. So it's a lot of interest to really understand better how this can be avoided and to develop models which can help in understanding this aspect. So one potential reason for drug failures is that you need to get enough of the drug into the solid tumor, if it's a solid tumor cancer, and throughout the tumor really to kill all the target cells. And of course, there are known differences in tumour physiology compared to other tissues, such as abnormalities in the tumour vasculature, and also the extracellular environment and also heterogeneity within the tumour, which can impede drug delivery to tumour cells. So as an example of some of these features are shown in the figure on the right. So for example, changes in the extracellular matrix compared to normal tissue, so this messy kind of environment shown in the figure can impair drug delivery, particularly of macromolecules, also potential for differences in drug binding, for example. And these really key barriers in terms of drug delivery can differ for small molecule drugs versus therapeutic proteins because of the different properties of these drugs. The much larger size of therapeutic proteins can affect the factors which we needed to consider really when developing these models. So the new features which will be included in Simcyp version 18 are a solid tumour model to describe the distribution of small molecule drugs, which will be available for both human and mouse simulators because of the interest really in modelling tumour mouse xenograft data and understanding potential differences between mouse and human. So the second development is the development of solid tumour models to describe the distribution of therapeutic proteins. In this case, this is only available for the human simulator simply because we don't have the large molecule models at all currently built into the mouse simulator we have. And for both these models, we've built an integration with tumour growth inhibition models. So you can account for the change in the tumour size and potential changes in other properties such as transporter abundance, for example, as the tumour grows for quite long-term experimental or clinical studies and incorporate these in to see the impact on drug distribution as well as being able to model the tumour growth itself. And this is incorporated to account for both single and combination therapy effects on the tumour growth inhibition. So focus first on the distribution model for small molecules. So the figure on the left shows the minimal PBPK model that we currently have in Simcyp for small molecules, and on the right-hand side, the full PBPK model, which accounts for distribution to many different organs. And it shows you how the tumour model has been incorporated within this current model setup. So it's linked to the systemic compartment, so this is described as plasma concentration of the drug, and that's linked to the tumour model as a distribution model for the minimal PBK model. You can also model the tumour model as part of the full PBK model. And in this case, we've considered separately either a lung tumour or a generic tumour. So the lung tumour is shown at the top of this figure, and this differs really just in terms of the connection of the blood flows. So for the lung tumour, it receives blood from the vascular circulation and returns blood to the arterial circulation, whereas the generic tumour is in parallel with most of the other organs, so it receives blood from the arterial circulation and returns blood to the venous circulation. So the tumour model is available for small molecules for the human simulator for substrate, inhibitor 1, and the primary metabolite of the substrate, so through 3 different moieties to model combination therapy, and for the mouse simulator for the substrate and the substrate primary metabolite. And again, this is down to differences in the mouse simulator and the fact that we don't currently have inhibitor models for the mouse simulator. So just to show you now the tumour distribution models in more detail on the model that we've included. So we're using a permeability limited distribution model structure, which has a number of similarities to the model that's currently available for the liver in Simcyp. And it makes a number of assumptions. So the first is that the unbound unionized drug is in an instantaneous equilibrium between the vascular space and the extracellular water. It can account for both passive and active movement of drugs across the cell membrane between the extracellular and intracellular compartments. So that allows the incorporation of active transporters as well as passive permeability. Can account for clearance of the unbound drug in the intracellular space, if there's known enzymatic degradation, for example, as well as non-specific binding to neutral phospholipids, neutral lipids, and acidic phospholipids if relevant. And these, again, can be predicted based on the tissue distribution models and the drug properties. And you can also account for specific binding to the target in the intracellular compartment. So this is something that's not incorporated in pre-existing permeability-limited distribution models, such as the liver model we have in Simcyp. Those are specifically of interest to the tumour, since target binding can potentially be quite significant. And as part of building of this model, we've collected data on the physiology of the tumour. So, for example, it's known that the extracellular pH is often slightly acidic. So we've incorporated these data as much as is available. In doing this, we have had to incorporate data from a mixture of different solid tumour types because of limitations in the data available. So we would encourage you, if you have your own data, to incorporate that as well for a specific tumour type of interest. Okay, so move on now to talk about the distribution model that we have for large molecules for the tumour. So again, just showing you on the right the models that we have available. So a full pubic decay model, also a minimal pubic decay model for therapeutic proteins. And these are quite similar to the small molecule models, but particularly for the minimal pubic decay model, there is some difference in terms of incorporating the permeability-limited distribution to the tissue, specifically for the therapeutic proteins. So again, similarly, the tumour models available for both full and minimal pubic decay models, both for monoclonal antibodies and antibody-drug conjugates, and also for other proteins and peptides. And similarly, we can count for lung tumour and generic tumour for the full pubic decay models. So in Simcyp, we do have separate models at the moment for monoclonal antibodies versus other types of proteins, just down to some of the differences in the key mechanisms involved for these different types of proteins. But the tumour model is available for both of these. So the tumour distribution model for large molecules is available only for the substrate at the moment. And also, if you're modelling an antibody-drug conjugate, we're able to model the formation of the payload within the tumour and the distribution of the payload that forms in the tumour using the small molecule model for the tumour that I've already described, and specifically accounting for the release of any drug in the tumour through target internalization and the TMDD mechanism, or through deconjugation, for example, within the tumour. In terms of the details of the model that's been included in the key mechanisms that are incorporated, this is the tumour distribution model that we have for therapeutic proteins. It's a permeability-limited three-compartment model, so it has vascular, interstitial, and intracellular compartments. Movement between the vascular and the interstitial space can be described via convection and diffusion. So there's quite a lot of evidence that in the core of the tumour, because of high interstitial pressure, there's an impairment of the lymphatic system and collapse of the lymphatic vessels. So based on this evidence, by default, we assume within the model that the lymphatic flow is zero. And you can edit this and modify this to investigate if there is lymph flow, but by default, we assume that that's not the case. And this means that when we make this assumption, that what happens is that lymph flow and therefore convection is no longer an important mechanism for the movement from drug from the vascular to the interstitial space, and movement is instead driven by the passive permeability or the diffusion into the interstitial space. And we've incorporated as part of the model a prediction method based on the published method of Schmidt and Wittrup in 2009 to predict the distribution and the passive permeability of proteins of different sizes from the vascular to the interstitial space. So that's an automated calculation within the simulator. You can also model target-mediated drug disposition as well as intracellular distribution through user input parameters. And the model, like all our therapeutic protein models, can account for distribution of both the unbound and the bound drug, and the impact that this has on the overall protein size, which can impact on the distribution. We've also accounted for the potential for exclusion of macromolecules in the tumour interstitial space, and this is really due to the changes in the expression and composition of the interstitial environment, for example, increased levels of collagen and an impaired structure really in terms of the arrangement of different fibers within the interstitial space, which is less organized, creating a sort of gel-like structure, which is actually a barrier to distribution of larger molecules. And again, this can be predicted based on the protein size and knowledge of that structure, and we've implemented a method to predict that as an automated calculation, again based on the publication by Schmidt and Ritter 2009. And the model that we have for monoclonal antibodies and ADCs, so this has many similarities with the general therapeutic protein model that I've described. The change really is it's still a three-compartment permeability limited model, but here we're considering specifically distribution to the endothelial compartment, while assuming that there's no distribution to the intracellular space. This isn't really relevant for very large therapeutic proteins such as monoclonal antibodies. So we need to model the endothelial compartment to model the SDRN protection mechanism. So that's incorporated as part of the MABS and ADC model. And you can also model target-mediated drug disposition within the interstitial compartment. For the ADC model, we can still model multiple ADC drug-antibody ratio species linked by the deconjugation and the formation of the payload within the tumour, which is then in turn modelled by the small molecule tumour model. And similar to what I described for the other therapeutic protein model, we can account for the steric exclusion of macromolecules, which is relevant really to monoclonal antibodies because of their large size and really impairs access to the interstitial space. So the third features the implementation of tumour growth inhibition models, and there are really three options available for modelling tumour volume. So if you're only interested in drug distribution in a short-term experiment, then you can just assume that the volume remains constant, so a static tumour model. Or we have either a built-in tumour growth inhibition model, so there's just a single option that's available at the moment, the widely used Simeone model. And you can also define your own tumour growth inhibition model via Lua custom model option that we have, which I'll explain in the next slide. So the built-in tumour growth model, I'm sure many of you are familiar with the Simeone model, which is illustrated at the top of the slide. So this assumes an exponential followed by linear growth phase of a tumour, and the drug effect comes in through a killing effect that's proportional to the concentration of the drug. I think one of the things that's different about PBPK models is that we're able to model the concentration in the tumour, and therefore we can use this as a driving concentration for the drug killing effect. So the drug concentration in the interstitial compartment of the tumour or the intracellular compartment of the tumour, and link this up as a site of action to the tumour growth inhibition. And then, of course, we've got the delay terms for the killing of the tumour and the damage to tumour cells and subsequent cell death in the model. So we can also account for combined drug effects. So for the built-in model, this is incorporated quite simply through a additive term and an interaction scalar, as shown at the bottom right of this slide. But you can incorporate more complex mechanisms through the Lua modelling option. So the custom tumour growth inhibition really allows a lot more flexibility. So it uses a Lua interface, which is similar to a PD custom option and granular growth model options, and a number of other options that we already have in Simcyp for those who are familiar with these. So it's basically a scripting option which allows you to write your own equations. And there's also options to call in certain parameters set by the screen, such as the initial tumour volume or the blood flow or any of the tumour growth inhibition model parameters or any of the concentrations from the tumour, also the plasma concentration as well. So these can all be used within the tumour growth inhibition model, which you can define yourself. And through this model, you can then define the tumour volume, but you can also define and modify certain other parameters of the tumour distribution models. So, for example, transporter expression, which you can change over time, or target expression, to incorporate these additional mechanisms and additional changes which may occur as the tumour grows and develops, or shrinks, of course, with growth inhibition and killing of the tumour by the drug. So this enables incorporation of much more complex scenarios. Okay, just finally to acknowledge my colleagues have also done a lot of work and helped in the development of this model. So this work was supervised by Lin Zong and In, and Khaled, Devon, Con, Isha, and Siri all contributed to the development of these models. Okay, so thank you for listening. I'll pass you on now to Shriram, who will introduce the final aspect of the webinar.
Yes, thanks, Rachel. The final aspect of the presentation is about the food staggering model, what we have got within simulator in version 18. So it's about expansion of our trial design to handle the food and dose staggering within Simcyp Simulator. So as many of you are aware of that in simulations, in Simcyp Simulator, you can simulate either a fast state condition or a fade state condition. But many of the times in real clinical protocols, generally, which looks like the schematic representation I am presenting here, is at least 10 hours overnight fasting volunteers enter into a clinical study. The dose is administered in the morning, and then after 2 hours, minimum after 2 hours, the volume is administered to the volunteers, and then 4 hours, minimum after 4 hours, the food is administered, depending upon the clinical protocol under consideration. This is about the fast state study. In case of PET clinical protocol, again, same as a fast state, the volunteers enter into clinical study with at least 10 hours of fasting, and just half an hour before the dose is administered, the volunteers are given food, whether it's a high-fat diet or low-fat diet. And again, same as a fast state, at least after 2 hours, the water is administered or given to the volunteers, and then 4 hours after the food is administered. So such kind of scenarios where food and fluid has been staggered with respect to the dose cannot be simulated till version 17 and before. Addition to that, even perpetrator could not be simulated or staggered with respect to the substrate dosing and that of the fluid and food staggering. So as a part of version 18, we have come up with the expansion of the trial design feature within Simcyp. So mainly the feature involves the 3 important aspects. The one is the expansion of the existing custom trial design for food effect evaluation, which basically allows the user to stagger the timing of the food as well as fluid with respect to the dosing, which is a substrate dosing or a perpetrator dosing. Additionally, you also user get an interactive trial design interface where they can visualize the trial design or the clinical protocol they want to simulate within Simcyp Simulator. The second important aspect of this feature is handling fast state and fade state simulations and vice versa. So this is another important aspect where we wanted the user to give a flexibility about staggering the food fluid as well as the food with respect to the dose. And once the food is given, all the physiological parameters will come back to the fast state and the vice versa. So handling of fast and fade state simulations dynamically was another important aspect or a feature of this food staggering wishlist item, which we added in version 18. The third important aspect is advanced dynamic modeling simulation framework, and this was again important because since we wanted to handle fast and fade state simulations, many of the physiological parameters like fluid dynamics, intestinal and stomach pH models, and also a bile source model was a static model. So in version 17 and before. So again, to handle the dynamism between fast and fade state, we have come up with an advanced dynamic modeling framework, which I'll explain in a couple of slides ahead. The final aspect, the whole project was just not about the modeling, but the team also took extensive literature analysis to build up the parameters within these dynamic models. So as a part of version 18, we reviewed and expand the physiological parameters in order to handle the staggering food effects project, which was a part of version 18. So as I mentioned, in addition to fast state and fade state, user will now get an additional option on simulator, that is food staggering. And once you click the food staggering option, the custom trial design pop-up will give you an option to define your clinical protocol. So as I said, if you want to define half an hour before the dosing, a high-fat fat can be administered. after 2 hours, the fluid can be administered to the volunteers, and then a minimum after 4 hours, again the low-fat diet, or depending upon the clinical protocol choice, the food can be administered to the volunteers. So these type of staggering food and fluid dosing regimen can be possible now in simulating in version 18 onwards in Simcyp. This is the second aspect of wishlist item, that we have now this trial design graphical interface, where the design what you have got within a food staggering custom dosing pop-up can now visualize within a simulator. So this interactive design will help to transfer these designs to your reports, which are being generally used for the regulatory submissions. As a part of dynamic models which are introduced in version 18, one of the most important, and to best of our knowledge, the first time a new advanced dynamic boil salt model has been added within Simcyp. So the model now allows to estimate the bile salts concentration, or predict the bile salts concentration in all the 9 GI compartments within ADAM model. So these are the time-dependent changes in the bile salts concentration, and the gallbladder emptying and the liver secretion of the bile salts directly into the duodenum has been now attached to IMMC, that is interstitial migrating motor complex. So the emptying of these bile salts from gallbladder into the first GI compartment, that is duodenum, will be different depending upon the IMMC cycle defined for each individual within a simulator. The other important changes which are occurring as a part of food staggering modeling within Simcyp is still now we had all the static secretions within a simulator for saliva, gastric juice, and pancreatic juice secretions. So when I say static, the values were same in a fasted and fed state. But now as a part of version 18, as I mentioned, to handle the dynamic transitions from fasted state to fed state, and then again returning back of these parameters back to fasted state, we have built up the dynamic models for most of the GI tract secretions. And one of the example which you can see on screen is a time-dependent saliva secretions. So as you can see, the saliva secretion increment in the rate has been connected to the eating time. So that's what the physiological data shows in a literature, that till the time the volunteers are chewing the food, the saliva secretion goes on increasing and reaches to its maximum rate. But the moment the chewing or the eating of the food is over, the saliva secretion comes back to the fasted state. So that dynamism of secretions has been inbuilt as a part of version 18 within a simulator. The other important aspect is a dynamic pH model. So the current radio telemetric capsules data shows that the pH in the intestine is quite highly variable in the proximal part of the intestine, which is basically a duodenal pulp and the mid-duodenum, compared to the distal part of the intestine, where the pH is quite compact in its variation, and it's been restricted to the narrow range. Addition to that, the pH is consistently increasing from the proximal part of the intestine to all the way to distal part of the intestine. So this time-dependent variation in the pH changes to simulate the proximal part of high variation, which probably may be due to the emptying of the stomach contents into the intestine, can be simulated within Simcyp as a part of dynamic pH model. So till version 17 and before, we had a static model for pH, but version 18 onwards, as I said, to handle the dynamic changes within the fed and fasted state, we have introduced this dynamic pH model in version 18. Similar to intestinal pH model, we also have a gastric pH model, and one of the important aspects of addition of this new model is we have got data now based on the FDA high-fat diet. So if the user clinical protocol involves administration of the FDA high-fat diet, user can select the new dynamic pH model within a gastric compartment, which has been built on the basis of FDA high-fat diet data from Coziole et al. Back in 2015. So this is a representation of the FDA high-fat diet-based pop-up screen within a Simcyp simulator. This is quite a busy slide, as you can see, but the main intention to present this slide is to show what are the new changes which are occurring or coming up as a part of version 18. As I've mentioned, users can have a staggering of fluid as well as food with respect to the perpetrator as well as the substrate. So staggering food effect has been added into the simulator. Most of the gastric parameters has been reviewed under the new analytical techniques available in recent times. So we reviewed the pH and bile source concentration data set within the stomach, as well as the colonic parameters for these physiological parameters. Then, as I said, the secretions within a GI tract, saliva secretion, gastric secretion, and pancreatic secretions are now depend upon the fasted and fed conditions. The dynamic bile model has been introduced, which will then attach to the IMMC cycles of each individual, which is simulated within a simulator. And as an artifact of changing of fluid dynamics, GI tract secretions, now we can have the time-dependent changes in the bile source concentration as a part of dynamic bile salt model, and time-dependent pH changes, which are occurring in the GI tract as a part of a luminal pH model, which was introduced. So this dynamism, which has been built as a part of version 18, we actually think is very important to simulate the actual physiological conditions, and also most importantly, the impact of fat content within a diet has now been introduced as a part of version 18. The couple of other things which I want to mention as a part of this webinar is the whole project was just not about the models, as I mentioned earlier, but also extensive literature analysis we did basically in the context of new analytical techniques available to pick up the new physiological values where patients or volunteers are actually in ambulatory conditions, which are more physiologically relevant rather than in clinic patient values. So these new data has been built up for all of these dynamic models within Simcyp. This is probably my last two slides. This is just an additional slide which shows what are the new additions which are coming in version 18. Recently, we have been a part of FDA grant for precipitation handling within Simcyp. And as a part of that grant, now new nucleation and crystal growth model has been added in version 18. Particle population balance model is a new addition for handling particle size distribution within Simcyp. And as a part of this particle population balance model, user can actually import the Malvern PSD data directly into Simcyp to do a multimodal analysis for different solid states. So now user can also simulate the crystalline as well as amorphous solid states within a Simcyp, which is, to best of our knowledge, one of the first simulator which will be handling these two solid states, not only at a population representative or a single individual level, but also at a population level. Excipients effects in terms of cyclodextrin and the solubility changes or free fraction changes with time are the another additions as a part of FDA grant. And finally, we also now allow the user to input IVIVC tools for supersaturating drug products, or which we call as non-monotonous dissolution profiles within Simcyp Simulator. The last slide, this slide shows in addition to the ADAM model changes which are occurring in version 18, we are now coming up with a virtual bioequivalence module within Simcyp, by which the user can able to simulate the two basic bioequivalence trials designs, such as crossover and parallel, for the population under consideration. It's a sim-healthy volunteers or the patient population under consideration, and then user can establish a bioequivalence between the substrate test compound and that of the reference compound. Going forward, the virtual bioequivalence module will be more advanced and will able to accommodate the newer trial design techniques, such as a scaled average bioequivalence, and also user can able to simulate bioequivalence between the two different populations, let's say extensive metabolizers against poor metabolizers, and against healthy volunteers against patient populations. So it's a combination of a substrate in different population files, and also the combination of different substrates, reference and test in the same population can be possible going forward in for virtual bioequivalence module. But currently in version 18, user can do the two basic trials, crossover and parallel. So this is the team who has been working on version 18 food staggering, as well as FDA grant project, which involves Konstantinos, Krishna, Oliver, Ramki, Janak, David, and Masood are the supervisors. And we also thank FDA for the grant, which we are working for precipitation. Thank you so much. So if you have any questions, we'll be happy to take them.
Okay, awesome. Thank you so much, Shriram and Nikunj and Rachel. As Shriram mentioned, we'd be happy to take your questions now. Just enter them into the Q&A box on the lower right. Please do direct them to all of our panelists. Looks like our first question for our panelists is for Rachel. And the audience member would like to know, how do you account for the number of tumors in the body, variability in tumor size, and the metastatic nature of tumors in your model?
So at the moment, we don't account for multiple tumor sites. It's just a single tumor, and that's all that you can model within the current model setting. So I suppose we don't account for multiple tumor sites. You can account for variations in tumor size. So there's simply a CV on the tumor volume, which can be incorporated. And also, I think with the lower scripting option, you could also start, if you wanted to relate parameters to tumor size, you could use that option, and some of the options we've got for feedback incorporated in with that modeling option to account for variations in tumor size. So that is something which could be considered. In terms of the metastasis nature, again, I think this is something which is a bit beyond the scope of the current model in terms of accounting for metastasis. It's more focused on drug distribution in a pre-existing solid tumor. It's not really going into that kind of detailed mechanism at this stage.
We've got a question here for Shriram. Shriram, can we conduct food staggering simulations in the pediatric population?
Yeah, that's a good question. So at this point of time, in version 18, basically we work on adult population within a simulator. So we haven't looked into a pediatric population. So the simple answer is no, not at this point of time. But definitely going forward, since the framework is at place now in a simulator, we will go back into the literature and see how much quality data which will be available in pediatric population to use the same framework within Simcyp.
Question here for the audience for Nikunj. Is the Luminal model in the Simcyp Simulator capable of performing bioequivalence assessment of locally acting drug products in the GI tract?
Yeah, I think that's a very good question. So I think the Luminal model is available to use for prodrugs which produces the metabolite that are locally acting, for example, sulfasalazine, which produces 5-aminosalicylic acid. So I think we are working on a case study at the moment where we would try to explore the new features to be able to simulate this type of prodrug scenarios as well as some of the metabolites that is converting back. The whole model is available to be linked to the tissue, so I think you would be able to explore not only the luminal concentration but also the enterocyte or colonic concentration if needed. So yes, in general, the model is fully capable of doing it, and I think we are looking into the examples to show the utility of the model in those areas.
I think we've got another question here for Shriram. Can we predict food-drug interactions, particularly the effect of food on metabolic enzymes?
So as far as the food staggering model is concerned, the predicting the food-drug interactions, the user has to define the interaction parameters within simulator, but at this point of time, no, you can't. So if the drug is getting degraded within the food components, then user definitely can add degradation rate constant within a Simcyp simulator to handle the impact of food. But at the moment, the framework is more about handling the impact of food ingredients on the secretions and the pH and bile source parameters. But its impact directly on the drug is depend on case by case basis what data is available, and that needs to be filled into simulator to build up.
Got another question here for Nikunj. Does the Simcyp simulator allow you to simulate the pharmacodynamic response of luminally formed drug metabolites?
Yes, so I think the Luminal model is fully connected to PD, so the user would be able to link both luminal or enterocyte concentration in all segments of GI tract for the parent as well as metabolite. So for example, if your metabolite is active drug and the product was given, then you can link the concentration to see the PD effect. And potentially it can be used to do therapeutic equivalence. So if the PD response is quite similar, but then user has to put their own PD model in. So there are series of models available, but the user has to parameterize it. So in general, yes, both substrate and metabolite, both luminal and enterocyte concentrations are available and fully linked to the PD model.
Okay, it looks like we've got one final question, and it's for Rachel. I understand that the Simcyp Simulator has a virtual cancer population. Can this population be used with the tumor model you've described?
Yeah, absolutely, and really we recommend using the cancer population with the tumor model. So that population accounts for some differences, for example, in the protein, plasma protein levels and hematocrit levels. So yeah, that can be used. So I think one additional comment on this is that the cancer population model that we have at the moment doesn't account for any time-dependent changes, for example, like we would incorporate for the tumor growth. And that's really down to limitations of the data available to incorporate disease-related changes over time in the cancer population. But yeah, you can, of course, use that cancer population with the tumor distribution model that we have. And we would really recommend to do that to better